like Norm showing up at Cheers. I think just about everybody knows Scott. And we're really fortunate. I think he's going to go. He's the research team leader for the thoracic oncology program, and I think he's going to tell us what's new and exciting. Scott.
Good. Hello, everybody. Let's see here. So before I start, a couple of things. First of all, Dawn, I didn't realize you were such a powerful public speaker. So thank you. I mean, I can't really talk anymore. I'm gonna, embarrass myself. But and thank you for sharing with us. There's so many things I could say, but other thing was I'm going to say Amy, our radiologist, because Amy, I got to tell you that there are several folks in this audience who will say, did Dr. I won't even say the name because all of you will ask me, did Dr. R read my film? Because if not, I really need her to read my film. So that's Dr. R, everybody. So I thought today I'd talk to you about clinical trials, just go through what a clinical trial is. Many of you know what a trial is, how we develop them, and some of the trials that we have at Yale, and then talk about some of the research that we're doing and really what we call translational research, which means we take findings from the laboratory and we apply them to the clinic, to patients in the clinic, and then we take things from the clinic, we bring it back to the laboratory, and then we bring it back to the clinic, back and forth to improve the care for our patients. So I took this from the NCI, the National Cancer Institute site, which I'd recommend to anyone. There are a lot of resources. There are a lot of frequently asked questions that you could learn about, and just a couple quotes from them about what a clinical trial is. And clinical trials are research studies that involve people. Through clinical trials, we find new ways to improve treatments and the quality of life for patients with disease. And they go on to remind us that clinical trials are the final step in a long process that begins with research in the lab. It's not as though, these drugs are made and then boom, they go into people. For years, we research these agents in animal models and in other types of models, and in particular in cancer models. And if we see promising results, then we would bring it in to the clinic. So there are three types of clinical trials that you may know of. There's phase one, that's the first type, phase one clinical trials. And a phase one clinical trials is asking the question, is the treatment safe? The purpose of the trial is to find a safe dose. Okay, so you start with a very low dose. The first 3 patients who are accrued to the trial will get that dose. If there are no significant toxicities, another 3 patients will be treated with a slightly higher dose, and so forth, until you reach the maximum tolerated dose. Traditionally, these trials have accrued 15 to 30 patients, but there's been a change in that these days we don't want to wait for the next trial, the phase 2 trial. So rather than doing a whole nother trial, we do these expansion cohorts where we might treat 100 patients with lung cancer in the context of a phase 1 trial. So most of the trials, the phase 1 trials that we put our patients on, they aren't first in man, you're one of the first patients getting this treatment. There might have been 50, 60 patients who got the treatment prior to you. And that's the majority of cases. So a phase 2 trial traditionally was a trial that asked the question, does the treatment work? So you have a phase 1 trial, you find the safe dose, and then you go into a phase 2 trial, you treat maybe 50 patients with lung cancer, and if you see a good response rate, that warrants further evaluation. The purpose is to, spelling error, but the purpose is really to see if there is a treatment effect. But of course, in any phase trial, we're always looking for safety signals. Is there a toxicity that we didn't realize in the early development of this drug? This is something that's put into all of our trials. So then we get the phase 3 trial. These are randomized trials. So question here, is this treatment better than what's already available? So a generic clinical trial will be, say, chemoimmunotherapy is the standard first-line therapy. Well, we might do a trial where we do standard chemoimmunotherapy compared to standard chemoimmunotherapy plus another agent, maybe a targeted agent or something else. Sometimes that targeted agent will be a placebo. right? So you won't know if you're getting the targeted agent or the placebo, but everyone's getting at least standard of care. People are always concerned that they're going to get the placebo. We wouldn't do that to a patient because it would be unethical unless there was no standard therapy. And rarely there'll be trials like that. We don't have any trials like that, but that would be the other situation. And these trials can have thousands of patients because you want to establish a new standard of the care, and the FDA requires a certain threshold to get to if this is going to be approved. So there are many potential benefits and risks. Of course, the real benefit is this the next home run? Am I going to get a drug that looks really good in either laboratory or in phase 1 trials, and will this really cure my cancer? Will this really lead to long-term survival? Also, by participating in a trial, you play an active role in your own healthcare, and you're also advancing medical knowledge. And some patients and some people feel very strongly about that. There are potential risks. Well, maybe this treatment, is not better than standard of care. But again, usually, if you're getting a treatment in a randomized way, you're getting standard therapy with or without. And unfortunately, a lot of the standard second or third line therapies for lung cancer, they aren't very good. Our main concern is unexpected side effects. So we have a general idea of what to think, but you can see late side effects, which would be concerning. There's also the potential for many more visits, testing, and really cost. I have to get back and forth. What do I do with my kids? Trials. Generally, if you're going to have a trial of an experimental agent, the agent is provided by the pharmaceutical company or some other source, and standard of care costs of the trial. So say you get scans every 2 months on the trial, insurances are expected to pay that, and that's really set by Medicare, and insurance do follow that. So it's very rare that I'd have a patient where their insurance wouldn't cover the standard care costs, and the patient would be required to pay additional costs in the context of the trial. That said, travel, co-pays, parking at air rights, all sorts of things. And we try to help as best we can with that, but there are limitations of what we can do. So hopefully myself or doctor here or your doctor in the community will talk to you very early on about clinical trials, whether you have a stage 1, 2, 3, or 4 lung cancer. And if not, feel free to ask them, because that's why they're there. And I think a doctor would be excited if someone said, well, listen, are there any novel clinical trials? Even if they don't have trials at their site, they know how to find the sites that might have trials that make sense. And if you're one of those folks who knows much more than me about, a lot of things that I studied years and years about, because you spend hours going through Google and stumping me, with questions, you can go, there are different sites that you can go to, and one great site is this clinicaltrials.gov site, which is a search engine for all the trials that are available for you. And they tell you basics of the trial, where it's open, how to contact, and you can bring that information. You can talk to your doctor, you can call the sites directly. So I would definitely take advantage of that site. So there's so many trials that I could talk to you about today, innovative trials that we're doing at Yale. And I had to pick one, so I picked this one because I think this is really in the forefront. And this is what we call cellular therapy. And the idea of cellular therapy is you take an immune cell out of the body and then you make it better. You enhance it, you grow it, and then you put it back into the body so now it can effectively treat cancer. And we know that the immune system has incredible potential because patients who do respond to immunotherapy, they can respond with metastatic disease for years and maybe even indefinitely. So the first trials of immunotherapy for lung cancer about 9-10 years ago were patients, these were phase 1 trials in patients who had multiple prior lines of therapy. They probably, prognosis was maybe 3 or 4 months at that point. And they got this therapy, and here we are 10 years later with some patients who have no evidence of disease after only receiving a year or 2 of therapy. So that potential is tremendous, and we're seeing it's not just the miracle patient. It's 15%, 10-15% of patients we see with metastatic lung cancer who have 5-year survivals. Right? Dawn talked about that magic mark there. So anyway. This is cellular therapy, and basically the way we do it at Yale right now is we take out, so here you can see a patient with multiple tumors, we take out one tumor, And then we remove the immune cells that are in that tumor that specifically recognize that tumor, that are supposed to attack that tumor. But what happens is when they get to the tumor poisons them or puts them to sleep. So now they can't really do what they're supposed to do. So what we do is we take those immune cells outside of the body. The patient goes home, and for about 2 or 3 weeks, we take out the cells, and then we enhance those cells. We make them better. We rejuvenate them. And not only that, we multiply them over and over and over, creating a tremendous army of a patient's own immune cells that recognize tumor, whose only job is to kill cancer, okay? So then what we do is we admit the patient, and the patient gets some chemotherapy to create the perfect environment for these cells to get where they need to go and to do what they're supposed to do. And after a week of chemotherapy, we take the army of immune cells and we infuse it back into the patient, and then we give an immune stimulant to try to get those cells going even more. And so I think this represents a very novel approach, and this is only available at a handful of sites around the country, really even around the world. So we do this with a company called Iovance, but Yale now has the ability to do this, and a trial was recently started in melanoma where we create all these cells. For the trial that we have, we send the resection specimen to a special lab, and they pull out the cells for us. All right, so before moving on to research, I just want to emphasize that part of what we do, one of the main parts is protecting our patients. And this happens even before a trial is started, really initially with scientific review by whoever is sponsoring the trial. So if it's pharma or if it's the NCI, they go through the protocol that specifies what we're going to do, and then make sure it's safe for the patient, that it makes sense for the patient. Every hospital has an institutional review board where they have to pass the trial that you want to do, and if they feel that it's not in the patient's best interest, they will not allow it to be run. And of course, there's informed consent. Of those folks out there who've gone on trials, they know that it's a painstaking process, but there's a 20, 30-page document that you read through. We go through it with you to explain every single risk and potential benefit, and then we have you sign that. During a trial, we continue to monitor how the drugs are doing, how the patients are doing through our institutional review board. And there's additional a data and safety monitoring board that reviews data as the trial goes. And the purpose here is to minimize risk, to ensure integrity of data. And the data monitoring board will sometimes close a trial because they'll say, this does not make sense. It's not in the patient's best interest. So, turning to research, and a lot of us, Dan talked about this a little bit, Rick also, we do what's called translational medicine. As I said before, starts either in the lab or the clinic, but then whatever we learn, say in the lab, we'll go to the clinic, and then we'll say, take for example, there's a drug that is developed in the laboratory based upon some animal studies and some cell line studies, and it looks really good, so we bring it to the patient. And let's say 1 out of 10 patients responds. So we want to know why that patient responded. So what we'll do is we'll go back, we'll take some of that patient's tissue, and then we'll look at the tissue from that patient as well as the tissue from patients who didn't respond. We'll bring it back to the laboratory. We'll look at that tissue. We'll do studies on the tissue to try to understand who is the person, why is the person responding, so we can improve the outcome in other patients. Or for example, say a patient responds to a therapy and then say a year or 2 later develops resistance. We'll do a biopsy, and then we'll compare that tissue to the tissue that we have from before the patient started that treatment, and we'll say, what happened here? And if we can figure out what happened, maybe we can give another therapy that will overcome that hurdle. So to do this, you can't just take tissue and run studies. You need to have a protocol, and you need to have consent from the patient. So we created a biopsy protocol at Yale, a tissue, shall I say, protocol at Yale that allows us to go down to say Dr. DiBiase has to do a bronchoscopy on a patient because a patient's coughing up some blood. And unfortunately, sometimes that happens with lung cancer. So she'll take her scope, she'll go into the airway, and she'll see a tumor, and she will debulk the tumor. She will cut out pieces of the tumor. She might cauterize areas that are bleeding. And what normally happens to that tissue, it goes into a bucket, and it's processed in a certain way that kills a lot of the things that we want to look at. And this process is put into a wax block, and then it sits on a shelf in the surgical path for 10, 15 years until they store it or throw it away. So what this allows us to do is to go down to the bronchoscopy and take a piece of that tissue that Dr. DiBiase took out, and we can do all sorts of things with it. We can inject it into a mouse, and then we can grow it out in our mice, and when a mice dies, we cut into 4 pieces, we put in 4 more mice, and now we have a whole litter of mice, and we treat the mice with different agents to see if we can overcome cancer. We can also do other studies. We can pull out immune cells, try to understand the immune cells. There's all sorts of things that we can do. And it's not just limited to tumor. Say you have a bad toxicity, a rash. We might take a biopsy of a rash to understand what it is, but also we can do additional studies so we can maybe prevent that rash or treat that rash more effectively in the future. But we need to have a protocol to do this. We need to have a patient saying, I consent to allow you to learn more about my tumor to help science. You know, it could help you, but that's not why we do it. We do it to help science, and if we learned something, of course, we would use it. So through this protocol and the protocol prior to it, over the last several years, we have enrolled 550 patients. 409 patients have enrolled. We subsequently had an amendment that allowed us to look at patients who had died, but of the 409 patients enrolled, 210 patients we have fresh tissue, meaning that we went down in 210 patients down to the procedure, we collected tissue, and we did special studies on them. So I could spend weeks, going through what we've done and the discoveries that we've made, and maybe at another talk I'll do that. But I just want to give you a sense of what we do here. We work with many different labs at Yale. Yale's a great place. It's got incredible depth when it comes to bench research. And when I first started here 15 or so years ago, there wasn't much of this. And I think one of the great things about Yale is over the last 10, 15 years, Smilow grew, and the engagement from the scientists, the basic scientists at Yale grew. And now everyone wants to do work with us, right? Because we can give the clinical perspective, the scenarios, and we can also help to get tissue and to understand and do some of the studies with these different labs. So we work with many different labs, and any of these guys would love to talk to you about what they do with the tumor specimens or blood specimens or other specimens that you give us so we can learn about cancer, so we can improve the care of our patients. I said I was only going to tell you about one trial, but I'm going to tell you about one more, and then I'm going to end, just to give you a sense of how we can do this in the context of a trial, a clinical trial. So this is a clinical trial for patients newly diagnosed advanced non-small cell lung cancer. And these patients get a combination of 2 immunotherapies. One is called Nivolumab. You might have heard of Opdivo. Some of you may have had Opdivo. And the other one is Ipilimumab or Yervoy. And in this trial, we do a biopsy before the patient receives therapy, and then we do a biopsy on therapy. We also collect blood, sputum, believe it or not, stool as well, because we're learning a lot about what we call the microbiome and the organisms that live in the stool and how that might actually affect your immune system ability to fight cancer. So this trial is technically an industry trial, but we really wrote it with Bristol Myers Squibb. And Kurt Schalper, who is a scientist at Yale, leads the translational aspect. So all the different sites around the country that are participating, they send their tissue to us so we can understand who responds and who doesn't respond. And I will say one other thing that someone asked, I think you asked the question about smoking, and I want to just say that if we abolish smoking, which would be great, but let's say smoking was abolished, and we go 100 years into the future, lung cancer will still be a relatively common disease. It is not just a disease of smokers, okay? And 15, 20% of patients never smoke. So considering lung cancer is such a common cancer, if you took away smoking, it still would be a tremendous problem for us. So as you may know, November is Lung Cancer Awareness Month. And I'm here to answer any questions, and thank you all for coming.
You've been on one trial. Are you allowed to go on another trial?
So the way I look at it is, in a patient, say, who's going to go on a trial, and I personally think, all patients should be on a trial, considering the types of trials that we have. It's what I would do because it's the only way you're going to improve things. And considering you're most of these trials, you're going to get standard plus. But the way I look at it is we have 2 or 3 potential trials for you. And if the first one doesn't hit, you do the second one, you do the third one. There are different trials. And when we look at patients who have advanced lung cancer, and there are trials for stage 1, stage 2, but when we look at patients who have advanced lung cancer, there's 2 general paths that we take. One is we evaluate what we call targeted therapy. So some patients, the minority, but some patients, maybe 15, 20% of patients, we will find a molecular alteration that drives the tumor, that fuels the tumor, and that's the most important thing in the tumor. And some of you, I know, EGFR is one, ALK is one, ROS is one, MET is one. And so we have these targeted agents that really block that pathway. And so that's one, and there are approved therapies for some patients, but if they fail those or there are no approved therapies, our trials could focus on that targeted pathway. That's one path. The other path are patients that don't have a specific molecular alteration that we can go after. And that's really where we concentrate on immunotherapy, because we see responses to immunotherapy across the board. So what we'll do is maybe we'll try 2 immunotherapies, or maybe we'll try the cellular therapy. And again, patient goes on one therapy on a trial. If it works, great. If it doesn't, if a patient is healthy, there's always going to be another trial, as long as a patient is fit enough to go on a trial and a patient wants to. Some patients don't, they really want to concentrate on quality of life, and then we do that. Marty.
How do you determine who qualifies for cellular therapy?
So there are eligibility, and in that particular trial, which I hope to amend with the company, it's pretty lax in that you can have no more than 3 prior lines of therapy, okay? And you can't have major autoimmune conditions. You have to have a certain breathing ability, which you would have. And so most patients would be eligible, but that is a tough trial because you get tough chemotherapy, you get tough immunotherapy, and you're in the hospital for 2 weeks for that trial. So you would probably reserve that for someone who you think can withstand that treatment, whereas most other trials aren't. That's probably one of the most potentially toxic ones that we have. Any other questions?
Can, Dr. Genninger?
Yeah.
Can clinical trials be taken at other satellite Smilow clinics? For instance, I live in Westerly, and the Smilow clinic satellite just opened there.
So there are currently trials open at many of the care centers, the Smilow care centers, and we're trying to open more. Some of the more intensive phase 1-ish type trials, they're tough to do there because there's many ancillary services that you need. And so we have patients who may go on a trial out in Westerly, and then maybe the next trial might be at Yale, the Smilow at the New Haven campus, this one, because it's a phase 1 trial. But we're trying to get more and more trials open out in the community.
And just another question. Having been a patient fairly recently, since I was diagnosed in July, spoken to a lot of healthcare professionals, and there seems to be quite a bit of disagreement about sugar. And if it's a causation, is it, can it worsen your disease? I've been told by people up at Dana-Farber that that's a myth. Other healthcare professionals feel that that is not the case, that sugar can be provide a negative impact to your cancer.
I don't think there's any consistent, decent evidence that suggests that sugar is harmful or helpful for your cancer. And certainly something I hear a lot. I would eat what you want to eat and obviously eat a healthy meal and exercise and do all that. But I wouldn't alter your sugar diet. Personally, I wouldn't alter your sugar diet for that. Now, there's evidence, and we're blinded as clinicians, clinician scientists. We need to see a trial. 500 patients get a lot of sugar, 500 patients don't get a lot of sugar. Who does better? If you go to an alternative medicine doctor, they might have a special diet for you, and they might be onto something. right? And they have anecdote. And maybe if they've treated thousands of patients, maybe anecdote makes sense. But for us, we need more evidence. And over the years, you've seen diets that are good for cancer, that are bad for cancer. And right now, I would eat what you want to eat and just follow just general guidelines about eating nutritious. None of us want to eat too much sugar, right? So my son doesn't let me, if I have a beer, my son says, "That was 6 slices of bread, Dad." I don't know where he got that from.
Thank you very much.
Any evidence of using viruses to attack cancer and kill it outright?
So that we've been looking at that for decades, and as we get more sophisticated, what we can do to a virus, yes, a virus, what we call oncolytic viruses that you can inject into a tumor, or right now you're limited because if you can inject, we do this, we inject into a tumor and it can kill parts of the tumor. If you inject it here and it goes throughout the body, you might have some side effects, some issues. But we are creating viruses that will hone into cancer cells, and then they will do their damage in those cancer cells. So yes, there is a lot of interest in all sorts of different virus vectors to introduce things or to kill things. I mean, you can also give a tumor something that it doesn't have through a virus. That would be helpful, So.
The person takes part in another clinical trial that's maybe affiliated with Yale but not at Yale. Who oversees that?
So if it's at one of the care centers, it would
Like at another hospital?
either if it was at a care center or if it was another hospital, it's the doctor at that place. Although, if I knew that there was a great trial in Boston or New York for a patient, I would get on the phone, I'd call my colleague, and I've done this so many times, and they've called us, especially for immunotherapy trials, because we're very strong with that. But for example, EGFR mutant trials, I would say, I get on the phone, I'd say, Dr. Helena, what do you got? And then I would set it up. So we're all in it together, whether you're in Boston, New York, or Connecticut. I don't know about, out west, but certainly on the East Coast, we're one big family, and we're here to find the best trial for patients. All right, well, thanks everyone for coming. I'll see many of you in the near future.